Welcome everyone, good afternoon, and thank you for tuning in to today's webinar presentation. This is the third webinar out of a 6-part series that will run every Thursday until May 7th as part of our Forest Invasive Spring Series. To learn about the rest of the series, please visit www.forestinvasives.ca. My name is David Nisbet, and I'm the coordinator for the Forest Invasives project at the Invasive Species Centre in Sault Ste. Marie, Ontario. The Invasive Species Centre is a nonprofit organization created in 2011. We connect stakeholders, knowledge, and technology to prevent and control the spread of invasive species that harm Canada's environment, economy, and society. We build networks of experts and stakeholders to identify and act on priority invasive species. We provide funding, coordinate, and lead projects in natural and applied science, technology transfer, outreach, and education. And we consolidate and disseminate information to raise awareness leading to the prevention of harmful invasive species. If you want to learn more about the ISC, you can visit our website at www.invasivespeciescentre.ca. Our speaker today will be Dr. Dean Thompson, who is a research scientist at the Canadian Forest Service. Dean joined the Canadian Forest Service in 1986, where he conducts research spanning fundamental and applied aspects of environmental chemistry and ecotoxicology as they relate to sustainable forest management and protection of the ecological integrity of Canadian forest ecosystems. He has published more than 80 articles in the peer-reviewed scientific literature and holds cross appointments as an adjunct professor at both the University of Guelph and the University of New Brunswick, St. John. Working in collaboration with colleague Dr. Blair Halson, Dean was directly involved in the development of the botanical systemic insecticide TreeAzin as a potential tool for management of emerald ash borer. More recently, he has been supervising laboratory and field research aimed at evaluating the potential use for TreeAzin against the Asian Longhorned Beetle. This afternoon, Dean will discuss results of these studies and continuing research in the context of developing an integrated response strategy for this invasive wood-boring insect pest. If you have a question for Dean during the presentation, please enter it into the side panel of the GoToWebinar program, and we will do our best to answer as many questions as possible with the remaining time at the end of the webinar. At this time, I would like to turn the presentation over to Dean, and he will take it from here.
Okay, I'd like to start, I guess, by thanking David Nisbet and the ISC team more generally for the invitation to speak this afternoon in the spring seminar series. As was recognized, we're going to talk over the next 30 or 40 minutes or so about Asian Longhorned Beetle, which I would classify as posing as a massive threat to the deciduous forests of Canada, as well as northeastern United States, and talk a little bit about the response that we should be taking in relation to that, and particularly an integrated response that has multiple layers of approaches that could help protect against those threats. So I'd like to acknowledge the fact that the Canadian Forest Service has been involved in doing research on invasive insect pests for quite some time, at least a decade and probably quite a lot longer than that. And in the ALB file specifically, Dr. Jean Turgeon and his team have been actively engaged in developing science-based information and knowledge in relation to the threat and how to deal with ALB. So it's really Jean that should be giving this presentation, but unfortunately you folks are saddled with me for the next little while at least. So I'll do my best to try to summarize some of that science background information, more general background, and then, as was mentioned, talk about systemic insecticides, and particularly TreeAzin and its potential for use against ALB towards the end of the discussion. So the key message I guess I'm trying to send this afternoon is that in responding to a threat as important and significant as ALB poses to Canada, it's important that we take basically everything and the kitchen sink and throw it at that problem. we need to be, and I say that sort of tongue-in-cheek, but we need to be hyper-vigilant in terms of detecting the beetle as it might occur in Canada. Take very aggressive action in trying to prevent its introduction, and we'll talk about prevention as one of the key elements of the response. We need to do very early and aggressive activities in terms of eradication attempts, and we need to be prepared in the unfortunate event, if it should ever occur, that ALB becomes actually broadly established within the forest systems of Canada. And thereby, by being prepared, I mean developing management options that could be applied in an integrated approach to responding to that problem. So that's my key message, and if you got nothing else from this presentation this afternoon, I think that is the key message that I wanted to send. We're going to talk about Asian Longhorned Beetle, and I'll refer to it in short form, ALB, as we go through the talk. Most folks will be aware that Anaphylora glabripennis, as it's known scientifically, is an exotic invasive wood-boring pest. It's native to China and the Korean Peninsula. What you may not know is it's been introduced to North America multiple times, actually, in a whole variety of different locations through North America, both the East Coast and the West Coast, although certainly our attention has been on the eastern infestations and interception, and that's occurred since the 1990s. The introductions have been largely through wood packaging materials that are contaminated with the borers. And by those, I mean crates and pallets and that sort of thing that are sent together with imported materials by a ship or air transport, that sort of thing. Of course, when it's introduced into a brand new environment like North America, it has not had a chance to co-develop with natural predators or parasites or other enemies, and therefore its populations could take off quite significantly because of the lack of those types of natural controls over the population. It's important to know that most, if not, well, I'll just say most of the initial infestations or first infestations that were discovered in North America were actually made by members of the public. And so having an awareness of what Asian Longhorned Beetle looks like and what the infestation symptoms look like is a key element, because public in general, and certainly arborists and other folks that are working in and around trees all the time, can act as sentinels and really help in terms of first detections of the beetles, and understanding and seeing a beetle infestation is critical in that context, because the earlier we get in to do something about it in terms of eradication, the more likely we are to succeed. So most people will never see a larvae or a pupae, as in the lower left-hand side of the slides here, but may come across an adult. It's pictured in the center photo there. They, of course, are shiny black in appearance, and they have predominant irregular white spots, usually 20 or so of those white spots along the wing casings, long antennae, and hence the name, that are banded white and black. And there's not that many species that they could be confused with in terms of large larvae or large adults like this, but monocamus, or the pine sawyer beetle, is certainly one that could be confused. One of the distinguishing characteristics is that monocamus have a single white spot right at the base of the wing casings here, and less distinct and less number of white spots over the back. So it's fairly easy to distinguish with a little bit of knowledge. The bottom line is, if you see something like this, then we want to make sure that it's reported and try to confirm whether or not it is ALB. Another significant characteristic is shown in the right-hand photo here is the exit holes, which are quite large, about the size of a dime or just less than the dime size, and they would be scattered through the upper bole of deciduous trees, particularly maples, and are good characteristic examples of an infestation of ALB potentially. Again, if you see those kinds of things, they should be reported and confirmed by a professional. So I'll move to slide 3 here and talk about the ALB life cycle very quickly. In North America, the ALB can be either a 1 or a 2-year life cycle, depending on when the eggs are actually deposited and how many degree days there are, temperatures, quality of the host, et cetera, and so it could have either a 1 or 2-year life cycle. If we start with the eggs are laid singly in what they call oviposition pits on the tree branch or in the bark of the bole or on the bark of the branches. And although they're laid singly, each female can actually produce something like 35 to 120 eggs. It's a large range, but it depends on the quality of the host substrate, the tree species that it's feeding on, and the health of the beetle, and several other factors. Once the eggs hatch, the early stage larvae feed in the cambium, so just under the bark, in the phloem, xylem interface there. They may feed in that area for 2 to 3 weeks, let's say, as a general time frame, and then are developing in terms of their size and growth through the mid-stage and late-stage larvae. The late-stage larvae will start to burrow deeper into the heartwood of the tree and will actually overwinter typically as larvae within the tree. They then form the pupae in the following spring and emerge as adults by exiting through those boring holes that we mentioned as the exit holes a minute ago. The adults can emerge anywhere between mid-June to mid-August, so it's quite protracted, their emergence period. It's not like they all come all at once, and that is a real problem in terms of developing controls and also in terms of monitoring for the adults. The females, when they do emerge, have to maturation feed, and so they're feeding on the foliage and the twigs and the petioles of leaves in the upper canopy, and they have a particular tendency to feed along the main vein in the foliage, as well as on the petioles and in the bark of the small twigs associated with that material. And I'll just point out while I have this slide up that the adult and the early stage larvae are really the two primary targets that we can aim at in the sense of developing controls, either be they systemic insecticides or bio. Okay, in terms of the trees that are susceptible to ALB attack, in North America there are several genera in that regard and more than 20 different species. Of those, the primary tree species include members of the maple, birch, willow, and poplar groups or genera. But as you can see from that list in the bottom right, there are quite a number of different tree species, all associated with ALB and which are known to develop. Maples, of course, are particularly important because they tend to dominate the southern deciduous forests of Canada in both natural and urban forest canopies. An example, in Toronto, 42% of the street trees in Toronto are considered susceptible hosts, and the majority of those are actually maples. So you can imagine if the beetle became established in the city of Toronto and actually killed all of those susceptible host trees, that would represent a major impact on the forest canopy in the urban center, as well as costs associated with dealing with that. Maples and proximity to roads are known to be important predictors of potential infestations. In its natural environment, ALB tends to infest trees that are more open growing, like hedgerows and that sort of scenario. And of course, the roads could be a potential vector of the beetles. They do seem to have a preference for maples, but as you mentioned, will infest a whole variety of other trees as well. Once they do infest a tree, it's actually the feeding in the cambium layer, so just under the bark in that cambium phloem xylem interface, where the early stage larvae are feeding, that create these galleries. In effect, if there's a high enough infestation, it girdles the tree, preventing upward flow of water and nutrients, and kills the tree by that mechanism. As we mentioned, later stage larvae will tend to burrow more into the heartwood and cause structural damage that can also contribute to tree mortality and pickup of diseases and that sort of thing. So a whole variety of different host tree species involved there, maples being the predominant target, but willows, birch, and poplar also being important. And I raised that issue now extending to a little bit broader perspective. If we look at the forest regions of Canada and we focus on what forest regions are actually susceptible to ALB attack, we can see the deciduous forest in red, the Great Lakes St. Lawrence forest in purple, and the Acadian forest region in orange here. That entire area is dominated by tree species that are susceptible to ALB. Many of those stands, natural stands, and many of the urban centers in those areas actually have tree species that are predominantly that predominate in the canopy and are susceptible to ALB. And that's not only in eastern Canada, of course, but a particular focus that we have here for the moment. And I'll draw the attention to the fact that all of that susceptible forest region is very closely associated with the Great Lakes St. Lawrence Seaway and the eastern coastal ports, Halifax is an example, and so on, New York and Boston. Those are the primary areas of import, so these ports are accepting shipping of major amounts each year and each day, in fact, and that's the port of entry that is the most likely route of introduction for ALB into North America. And you can actually see that that's the case. The yellow stars denote the actual infestations that have occurred. Starting in 1996, the first detection of an infested area in New York, and progressively in Chicago in 1998, and so on with Boston and New York and New Jersey and Ohio, and of course the two infestations that have occurred in Canada in the Toronto area in 2003 and in 2013. And as we switch to the next slide, I'll just emphasize the fact that the number of imports that are coming from international global trade is, of course, increasing, and that includes from China. There's been some estimates of the increase in trade there rising from, say, I believe it's $5 billion to $70 billion worth of trade coming through these shipments from China. And that, of course, enhances the potential that there will be new introductions of ALB associated with that. And we'll get back to that when we talk a bit about prevention here. In terms of impact, should the beetle become established, the economic and ecological impacts of Asian Longhorned Beetle could be absolutely massive. There have been estimates associated with a whole variety of different things. I'll just point out the fact that we're talking in the realm of billions and millions of dollars annually in total cost in the billions. And when you start talking about those kinds of numbers, that's a significant impact by anybody's estimation. So, for example, Noah et al. in 2011 suggested that there might be $669 billion cost through the loss of urban trees in, I believe it was just 9 U.S. cities alone. The hardwood products industry in Canada is worth something like $11 billion annually. So even a small hit on that kind of magnitude would be a significant economic impact. And then we can add the maple syrup industry at $100 million a year, and the cost of eradication, which in the U.S. has been estimated just up till 2006 alone as being something in the order of $800 million. And then indirect costs, for example, associated with loss of tourism. As all of us will probably realize, Canada is an attractive country to visit, and most people coming are looking for the natural environment, including our beautiful forests, particularly in the fall. And if there's a hit on those kinds of forest canopies in our parks and our conservation areas and so on, then that's going to reduce the tourism value. And as an example, 11.9 million visitors to Canadian parks on an annual basis. And it's not only economic losses, of course, but also ecological losses. So where we have, should the beetle become widely established and where we would have infestations in natural forest canopies as like the one you see in the bottom right-hand slide here, virtually everything that you see in color there would be wiped out. And the loss of forest canopy, where those susceptible trees are dominant within the canopy of natural forest, are going to result then in loss of wildlife habitat. And that's particularly important in terms of species at risk, and in southern Ontario, which is a high-risk area, because most of our species at risk are located in that area, and also associated. Many of those are associated with fragment or small woodlots that remain in those areas, and many of those are dominated by host susceptible tree species. So we have impacts on wildlife potential for species at risk, and also on the general ecological function of those sites in terms of carbon sequestration, protection against erosion, and susceptibility to fire, and so on. So a combination of both economic and ecological risks associated with the impact of ALBs. Okay, so the first step in response to that huge risk and the threat that's posed by ALB is actually prevention. David, maybe I'll get you to go backwards on some slide there. Okay, so in terms of prevention, I guess one point that we should make is that there have actually been multiple interceptions of ALB in warehouses and so on as a result of inspections, as you see pictured in the center slide there. In fact, there have been 17 different states throughout North America where there have been interceptions, as well as in Canada, and not only in the Northeast, but also in British Columbia and California. And that has been occurring since 1992, so it's not like it's a one-off deal here. There have been interceptions in all of those states and in Canada, as well as in several countries in Europe as well, Italy, Germany, and Austria, for example. So it's not Canada alone or North America where we have this problem, but certainly we do have the problem in terms of the multiple infestation sites here. Ports of entry are always involved as a priority in terms of where these interceptions are occurring, and it's largely, as I mentioned before, with the solid wood packaging material coming in with shipments such as the one shown here on the left-hand side, photograph containing multiple containers. And if you've been around any one of these big industrial ports, you'll see hundreds of ships and literally thousands of containers, all of them with the potential to carry wood packaging material and infested wood packaging material. So the prevention part of this, of course, is doing the inspections, and of course it's impossible to inspect every single container on every single ship coming in or in aircraft transport. But they impose phytosanitary measures both in the U.S. and of course in Canada, and now there's an international phytosanitary measure 15 that requires heat treatment or chemical fumigation of wood packaging to try to prevent input of these kinds of insect pests. But that's not a perfect scenario. It's not an absolute guarantee that there's going to be no new introductions. And examples of that, Hack and Petrus in 2009 showed that even wood materials that have been treated by fumigation or heat treatment and are marked as such carry 1.2% of those actually carrying quarantined insects of interest. That doesn't sound like a very high percentage, but when you talk about the gazillions of materials that are moving in with these kinds of imports, then that becomes a significant, still potential route of importation of ALB, even though we have these phytosanitary measures in place. And in fact, Hack just recently estimated that on an annual basis in the U.S., there could be 13,000 containers containing live wood pests of interest in the U.S. So that's a good example of the kind of pressure that's there for trying to prevent that from occurring, and it's not a nice story in the sense of our likelihood of success there. There are also, of course, importation regulations prohibiting the movement or importation of logs and firewood from known infested areas by surface transport, logs moving by truck across borders, or from infested areas even within a state or within a province, of course. So that's the prevention story. I guess the simple thing is we're doing a great job on that. There's lots of regulations and rules in place. There's effectiveness in the prevention measures, but they're not 100% foolproof. In terms of the next phase, or the other integration of a layer of protection, is, of course, eradication. So should an introduction actually occur and become established as a localized population, then the next approach is to try to eradicate that population. And again, I'll reference the fact that first discoveries of many of these infestations are typically made by the public, almost fortuitously. But as we enhance our outreach and education programs and get people familiar with what ALB looks like and the damage it can cause and the importance, that will be a major positive in terms of early detection of these populations. And also for arborists that are working in and around trees all the time, their knowledge, of course, increasing all the time with regard to this threat, and that can also be very helpful in terms of early detection. Once infestation is confirmed, then detection surveys are imposed so that we have visual surveys with ground-based crews and tree climbers in the case of ALB, trying to basically identify where the infested trees are and determine where the source area is, and basically bound the area of the infestation. That has been greatly enhanced, the effectiveness of those surveys, by development of training guides, signs and symptoms. So, for example, the production of the training guide by Rick et al. From our own location here in Sault Ste. Marie. And also the fact that those regulated areas are established to minimize the potential for assisted spread of infestations through movement of contaminated wood materials, such as firewood, as you see in the bottom right here. So once the infestation zone, the quarantine zone, has been established, of course, there's destructive removal. That involves cutting the trees and chipping them to very small sizes that are considered incapable of supporting ALB. Not only are the infested trees themselves destructively removed, but also host trees within 400 or an 800 meter radius, depending on the situation, of those infested trees. So that can result in massive destruction of trees simply to try to control a local population. Seems very aggressive, but it is actually required, because the risk, if you don't take that kind of aggressive approach, is absolutely massive, as we described previously. So, for example, in the original infestation in Vaughan in 2003 in North Toronto, 30,000 trees approximately have been destroyed in that eradication effort, and similarly in Worcester County, Massachusetts, a similar number of trees. So the number of trees, it depends on how many are infested and how far they're spread, and it varies on the site-by-site basis. The eradication program always go forward with an outreach and education component so that the local populace knows about the regulations regarding movement of firewood and other contaminated materials, and also aware of what the problem is and how it's being dealt with. So that's the second phase. So we have now prevention and eradication, and then if the worst case scenario were to occur and we actually had a broader establishment of the pest, then we would be looking at potential mechanisms of management options. Those would include biological controls and or the use of systemic insecticides. So what you've seen so far is the prevention and eradication steps, and that's all that has been going on actually in terms of responding to ALB in an operational sense. There have been lots of research, though, going on in terms of developing biological controls and also systemic insecticides. And I'll go over some of that research so you have a background of information to know what we're attempting to develop in terms of management options. There are several different approaches to biological control. I guess as an overriding statement, what I would say is that these things take a long time to develop and test and prove that they're going to be effective and efficient if they were to be introduced against a pest like ALB. And typically they involve sort of conventional biocontrol approaches where they go back to the native range and look for natural enemies in its native environment. That has been done with ALB, and they found that, for example, 2 Asian parasitoid wasps that are known to parasitize ALB in China and the Korean Peninsula. And there would be potential to import these and use them in a biocontrol option in the U.S., but there's a lot of testing that has to go on to look at how efficient those parasitoids are in terms of tracking down ALB in low-level populations, and how broad the host range for the parasitoids would be. Would they attack other beneficial organisms, for example? All those kinds of things. So it typically takes multiple years, wild guess at maybe 10 years to develop a conventional biocontrol for these types of pests. They also look at natural native parasitoids in the new environment, so in this case in North America, and see whether or not those could adapt and utilize ALB as a prey. And so, for example, there have been 4 native North American parasitoid wasps. 2 of them are identified there. I won't even try the names. And these are known to complete development on ALB, at least under laboratory conditions. Whether that would occur in the real world, again, under different population levels and so on, would remain to be seen. But there's certainly great activity going on, largely with Mike Smith and his group in the U.S. Looking at these sorts of biocontrols. Another approach is the use of entomopathogenic fungi. In this case, quite an interesting one, where the fungi is basically impregnated in a fabric band that is deployed around the tree. And that fungus, there's been several tested. It looks like this metarhizium fungus is one of the ones that has the highest virulence against ALB. It's also got the benefit of being already registered for pest control in the USA. So there's potential for using that sort of an approach to kill the adults as they walk along and pick up the fungus from that fabric band. And the reason for the fabric band impregnation approach is that it enhances the longevity and the viability of the fungus through time, so increases the potential for infection to occur and kill the beetle. And we can use, there's been studies on nematodes as well that are known to attack ALB and mate mid to late instars and produce high levels of mortality. Of course, there are all kinds of questions with regard to how many of these kinds of tools would have to be imposed within a tree. How do you ensure that the infections actually occur, and how do we deploy these over a broader spatial area in a practical, economical sense, all those kinds of things. So lots of work still to do on biocontrols, but at least we know that there's activity going on in that area. And I'm going to actually skip over the next slide here that talks about resistant trees, but just mention the fact that resistant trees have been identified. There are ways that we could use those in restocking areas, and if we could understand the biochemical mechanisms, for example, in calorie pear that's resistant to ALB, and which prevents the development of eggs in adults and reduces adult longevity, there may be ways to use that knowledge in terms of dealing as another element of a management technique in an integrated pest management response. And now we'll move to insecticides, and particularly systemic insecticides, which have the greatest potential, I think, for use against wood-boring insect pests like Asian Longhorned Beetles. So imidacloprid is a neonicotinoid derived from nicotine, actually, a synthetic chemical that's been used, actually the only product that's been used operationally against ALB in USA eradication programs. The initial work on imidacloprid was done by Trees Poland in China and showed significant reduction in ALB densities in willow and poplar that were injected with imidacloprid compound. The residue levels that are expressed in the foliage and twigs are in the range of concentrations that would induce high levels of larval mortality or adult mortality on the basis of those studies. And there's been some background work on artificial diet done with that as well. More recently, Eugene et al., I believe from Cornell University, have been publishing on imidacloprid and emphasizing the point that the residues in injected Norway maple, so this is maple trees that were actually injected as part of the response to the Worcester, Massachusetts infestation, show some degree of variability of imidacloprid. That doesn't surprise me. We've done some work on ash in relation to EAB with imidacloprid originally and found similar sorts of variability. And so depending on the actual material you use and the formulation that's applied, you can certainly get these kinds of variations within the tree or actually very poor uptake in general. But with the right formulation under the right conditions, you see rapid uptake, typically of the residues following the stem injection, and they are expressed in relatively high levels. So it's seasonal means here throughout the summer. For this particular study, we're 0.9 to 1.2 parts per million. That might not mean much to many people, but it's in the range that we know will have effects on adults. So, for example, kill 50%. That kind of range of concentration would kill 50% of the adults with those kinds of exposures and result in, say, 94% reduction in larval production. So reducing the fecundity of those adults that aren't killed. So there's definitely evidence out there to suggest that imidacloprid is effective, and another example of that is work by Sawyer in 2006 that reported out of 250,000 trees that were treated with imidacloprid in 3 different states in the U.S. associated with these eradication programs, only 11 were found to be infested with ALB. So that shows about a 0.004% infestation occurring after imidacloprid treatment, and shows that it's highly efficacious in controlling ALB. We do have this issue with variability of residues and the potential that some of the area of the tree might not have a high enough dose to induce these kinds of effects. And another potential here is that we have undesirable environmental effects of imidacloprid. So people will be aware of the impact on pollinators and bee colony decline and that sort of thing, and the fact that neonicotinoid type compounds have been closely associated or implicated in those controversies. We know from our own studies, colleague Dave Kreutzweiser and I have done studies looking at the transfer or potential transfer of imidacloprid residues from treated ash and potential effects on aquatic and terrestrial decomposer organisms, and do show that the senescent leaf material that is excised from the tree in the fall could have impacts on decomposition of foliar material, which is a key element of environmental function. And imidacloprid is also known to be potential for leaching and movement into surface waters and have activity against aquatic insects in a general sense that way. So there's issues associated with that product. Of course, we have to balance that with the impact of losing all of those trees and ALB killing all those trees and the environmental impacts associated with a do-nothing approach as well, if we're not successful in any other way. So not only imidacloprid has been investigated, but there's been research done on a whole variety of other insecticides. I'll just try to touch on a few of those that do show strong potential. One of those is certainly emamectin benzoate. It's a product that was also investigated by Trees Poland originally back in China, and I'm waiting for my slide to come up here. Okay, it's actually an avermectin. Emamectin benzoate is an avermectin compound, so it's derived from natural bacteria, but it's synthesized So the identity of the first product was a natural compound, but it's been since modified and developed synthetically. But it shows high levels of effect on ALB in terms of 71% reduction in ALB density on stem-injected willow. And Deb McCullough, her group at Michigan State University, and Dave Smithley, been looking at emamectin for control of EAB and show effective 3-year control from a single injection of emamectin. So that definitely shows some promise, and I would suspect that we can see similar levels of activity on ALB. One aspect of this is that persistence, the 3-year control is a result of the chemical being more persistent, and that can have a double-edged sword to it. In fact, you get greater target effects, but there's also the potential for greater non-target effects over longer time periods if you have a persistent compound like that. And there's other neonicotinoids, including dinitrotoluene, that show activity, strong activity against ALB. That particular compound can be applied as a basal bark spray, and it penetrates the bark and moves up into the foliage of treated leaves, and at least with EAB, or emerald ash borer, induces high levels of mortality in adults that are feeding on foliage of treated trees. Lambda-cyhalothrin is another compound, completely different. This is a synthetic pyrethroid, and it's interesting in the sense they're using this band approach to impregnate the bands, apply the bands to the trees, and then the adults that are walking across these contaminated bands would pick up a dose of this pyrethroid. And it has a very rapid knockdown effect and very effective. It kills 100% of the adults that cross the bands for up to 90 days. So this is a tool that could be particularly useful with a trap and kill sort of approach, and I think that's the primary way that they're promoting it. So you would put out, for example, potted trees that are very attractive to the ALB, cover those with bands, or deploy the bands with those trees, and any ALB that's landing and walking around on those trees are then killed. So it could be a monitoring technique, and it could be used to reduce localized populations as well. And then azadirachtin, and so these are natural triterpenoid compounds derived from the neem tree, and the basis of the TreeAzin formulation that the Canadian Forest Service and Bioforest Technologies locally here in Sault Ste. Marie have been developing. First targeted for use against EAB and has proven very effective in protecting ash against the emerald ash borer, but now we're expanding that to look at Asian Longhorned Beetle. So I'll talk a bit about that as well. TreeAzin is a product that's been developed, as I said, between CFS, originally discovered by Blair Helson, and developed to a degree, and then we've licensed that technology to Bioforest Technologies, and they have the worldwide market for that product. It's been registered in both Canada and the USA. Depends on azadirachtin A and B, which are these natural compounds that are extracted from the neem tree and then reformulated and injected into ash for control of the EAB, for example. And so really what we're doing there is moving a natural compound from one tree to another with this injection technology. One of the beauties of azadirachtin and the reason why we focused on this in developing a tool against EAB was its low toxicity to mammals, the fact that it's non-persistent, but sufficiently persistent to induce significant levels of control of EAB and protect trees. It also has a relatively low toxicity to bees compared to other products. It's registered to be used only by licensed applicators. They use the EcoJect system. This is an advanced direct injection technology. You can see it pictured in the lower slide here. Involves pressurized containers that inject the tree straight into a hole that's drilled in the base of the stem. The chemical moves up with the transpiration stream in the trees and is expressed in the foliage and the twigs, and also in the xylem phloem cambium interface where it exerts effects on larvae. That injection technique is pretty important because it really almost eliminates any potential for other exposure to humans or other organisms. So only those organisms really feeding within the tree itself are going to be exposed to the chemical, and that has a great environmental protection component to it. So well suited to use in urban scenarios, for single street trees or in park settings, that sort of thing. We know from our studies on TreeAzin going back to 2003 that it's very highly effective in controlling growth and inhibiting the growth of larvae that are feeding in that cambial surface area just under the bark of ash trees. And in fact, any exposure of significance to those larvae will basically shut them down in terms of their growth and development. We also have demonstrated, although not published yet, adult fecundity effects. So we know that TreeAzin, where adults are maturation feeding EAB adults, that is maturation feeding on contaminated leaf material, will have a strong effect in terms of their ability to produce live eggs, and that fecundity effect is dose dependent, with a greater number of eggs being produced at low doses, but as you go up in the dose range to anything of realistic, what we see in terms of realistic dose levels in the foliage, they have very significant effects on their ability to produce eggs and thereby contribute to future population. So in terms of uptake and translocation of TreeAzin, one of the first, so now we're trying to move from the success story on EAB and hopefully get that to occur also with ALB. And this involves the need to look at uptake and translocation of the chemical in susceptible host tree species. So we did a study in Oakville actually on this with 4 different species: sugar maple, Norway maple, London plane tree, and red Grimani maple. All typical sort of urban environmental trees. And in 3 of 4 of those test species, we see that same sort of rapid uptake and translocation into the canopy, well distributed through the canopy, maximum residues in less than 2 days post-treatment, and foliar residues in the 1 to 5 part per million range, with twigs having somewhat less concentrations, but typically in the 0.25 to 1 part per million range. And those residues, although they're non-persistent, do last for up to 20 days above the 0.25 part per million threshold, and that's the threshold that we believe is representative of significant biological effects based on our lab studies. So we know that the chemical moves well in maples and susceptible trees, with the exception of sugar maple so far, which tended to be the exception in that study, and showed quite low, exceptionally low uptake actually, and we think that this might be an artifact of conjugation of the chemical with sugars, and we're pursuing that and this year actually, and hoping to demonstrate that even though we can't see it analytically, that it actually still has biological effect on organisms feeding in sugar maple. And we'll know that is quite important. We'll know that after this year, I hope, and it's quite important in terms of protecting sugar maples, and particularly with regards to the maple syrup industry and its potential effects on those types of trees. From our laboratory studies, these are conducted on ALB in the Invasive Species Center quarantine lab, and the Invasive Species Center has been very supportive of our work in this regard, both providing space in the quarantine area and also financially supporting this area of research. So we've been doing larval experiments and adult mating pair experiments with TreeAzin against ALB specifically. The larval experiments involve dosing the artificial diet and allowing the larvae to feed on that diet and therefore be exposed to TreeAzin. And where we've looked at early, mid, and late stage larvae to see if there's a differential response at different growth stages, or different developmental stages, I should say, of those larvae. The primary response variable being growth inhibition, so we're looking to see whether it inhibits the growth. And I should use this as a point to emphasize the fact that azadirachtins are not acutely toxic like the neonicotinoids or pyrethroids. They don't kill outright. They actually kill by growth inhibition, so the larvae effectively starve to death and fail to develop through their normal pattern for the pupal stage. And another effect that they tend to have, those compounds, are inhibition of fecundity or reproductive success. So in that regard, we look at adult mating pairs where we load a stem or a twig of a susceptible host like striped maple, allow the adults to feed on that material and for them to mate, and then move them on to clean striped maple logs and then allow them to oviposit and measure the number of eggs produced, so looking at the number of viable eggs. And what we see in essence is a very similar sort of trends that we saw with EAD. The larvae are strongly inhibited in terms of their growth inhibition, so ALB larvae exposed to TreeAzin at significant concentrations certainly halt their growth for quite a significant period of time. The longer the dose and the greater the concentration, the greater the effect, and at 5 parts per million, for example, effectively shuts down any growth whatsoever. If the doses are lower, we get a lower period of time where they have no growth, and they will eventually recover if they're put on clean material, but they never do acquire the same level of size that the controls do. And that's quite important because we know from other work by Melody Keenan and others that a critical mass is required for pupation to occur successfessly. And so if we can inhibit their growth of the larvae in the treated trees for a long enough period of time to a high enough level, then it's likely that they would pupate and contribute to the population. Certainly they'd be feeding less and doing less damage and thereby protecting the tree. And we've established 0.25 parts per million as a threshold of significant biological activity here. So if we have those kinds of concentrations in the trees, which we do have based on our Oakville study, then we would expect significant inhibition of larval growth under those conditions. And we know that early larval stages are more susceptible than later larval stages. So the earlier we apply the treatment and target those early larvae that are feeding in the canidium phloem, then that's the most sensitive stage and are most likely to succumb to these effects. In terms of a visual representation of that, if you're looking at this slide, you can see the upper photographs are of a control over 70 days for early stage or 2-week-old larvae. They start out very, very tiny, but they grow incredibly quickly and get to be quite large larvae after a period of 7 days. But if they're treated with any significant level of azadirachtin or TreeAzin, you see essentially no growth there. Similarly with mid-stage larvae, so if you start with a later stage larvae, they also of course grow dramatically in the controls, but almost not at all in the treated levels, at least if the exposure concentrations are in that 1 to 5 parts per million range. Moving just to adults here and starting to tie off, I'm going to skip that slide just for the sake of time. The message here is the longer the exposure period, the greater the effect, and we know that we need at least 3 days of residues in the trees in order to get that growth inhibition effect. And the longer we have it in there for 20 days, which is what we would estimate based on our study in Oakville, the greater the period of no growth, and we would estimate 7 weeks in that context. Sorry, so moving to adult exposures here. Again, we've seen the same sorts of things we've observed with EAD. If we expose adult mating pairs to even very dilute solutions of TreeAzin through the systemic loading of twigs and allow them to feed on that, we see dramatic reductions in the number of cumulative eggs of live eggs that are produced relative to the controls. And we also see significant reductions in the female effort to oviposit based on the number of oviposition pits that they scrape to put their eggs in. And you can see one of those oviposition pits in this slide here, where the adults are mating, and she will then place her one single egg in that oviposition pit once they're done having fun there. And I'll point out Amanda Tonin, who's been the technician involved in doing all of this work on TreeAzin versus ALB. She's done an excellent job and responsible for all of the data that you're seeing there on ALB. An example of one of our test studies in the controlled chamber there in the IFC quarantine lab. So in terms of TreeAzin, we know that it's taken up rapidly and translocated well in susceptible tree species, with the exception of sugar maple. We're pursuing that unusual scenario there with sugar maple this year. Residue levels are at sufficiently high concentrations and sufficiently long duration to induce biological effects on ALB based on our Oakville study. And in the laboratory, when we look at the kind of effects that we see under those kinds of concentrations, we would predict significant inhibition of larval growth and also impairment of fecundity. So this is the so-called double whammy effect again, where we're influencing not only the larval life stage, but also the fecundity of adults when they're exposed in trees. And we would expect then under an operational scenario that we would get both those effects and protect the tree as well as reduce local populations of the ALB within that tree. So overall conclusions, say that ALB poses a massive risk both economically and ecologically to the deciduous forests of Canada. That we need to take a very multi-layered, integrative approach and basically throw everything we have at it to protect against that threat, including preventative measures, reducing the risk. We know that those phytosanitary measures are effective, and we need to keep using them and developing even better tools like that to prevent new introductions. But we know that with increased global trade and the fact that those preventative measures are not 100% effective, that there is very likely to be new introductions, as we've seen through the last decade, into North America. Eradication has been successful in some cases. There have been several in the U.S. now that where eradication has been declared and no new finds found in those areas. So we know that effective eradication techniques, and they're getting better all the time, can be successful. They involve the need for intensive monitoring and very aggressive response in terms of destructively removing trees and reducing that population in the infested trees, and protection in terms of destructively harvesting susceptible host trees in that area as well. And finally, should both of those techniques fail, then we need management options that allow people to protect trees and try to reduce the spread of ALB and these kinds of wood-boring insects. That should include the development of systemic insecticides and biological controls as well. And there's lots of good research going on in that area, some of which shows pretty significant promise. We need to get those to completion so that we're ready should this beast ever actually become widely established in Canada. And if that, I'll end off and hopefully take some questions here. I'll leave the acknowledgement slide up for a moment just to ensure that people realize that this is a multi-agency initiative involving the Canadian Forest Service, the CFIA, the Ontario Ministry of Natural Resources and Forests, and a variety of academic institutions, the town of Oakville, Bioforest Technologies, and the Invasive Species Center. And it's really that collective investment of time and effort and money that's required to deal with a pest like ALB. And with that, David, if we want to take some questions, I've certainly got time, and hopefully other people have the interest to do so.
Yeah, for sure. Thank you very much, Dean. That was a great presentation, and I'm sorry for some of the lag time you were experiencing throughout. But we do have a few minutes for questions here. So if someone has a question for Dean, you can enter it into the question panel on the side of the program, and we'll try and answer as many as possible with the time remaining. Dean, the first question I have for you here, is there evidence that Asian Longhorned Beetle will move out of the urban areas and into natural forests?
Yeah, the simple answer to that is yes. A little bit more detail. In the natural home range, ALB tend to infest, as I think I mentioned, hedgerows and more open-growing trees, and that's certainly been true in North America as well. But For example, in the Worcester and Claremont, Ohio infestations, there is evidence that they are actually moving into more natural woodlot types of environments, so multi-species stands, and that actually was the case in Vaughan as well. Although the infestations there tended to be on the periphery of those woodlots, there is indication that they will move in now into those woodlots and infest trees within those stands as well.
The next question I have here, why has the emerald ash borer managed to get as far as it has where the Asian longhorned beetle has been declared eradicated a few times already?
Okay, that's a complicated, a much more complicated answer that I'm really not capable of answering fully, but I would say that EAB, there's been a large degree of movement with infested wood materials, so a lot of there's been surface transport that's certainly human-assisted and enhancing the ability of that beast to spread. I'd say it's fair to say that the initial reaction to EAB was much slower and much less aggressive than it could have been, and a slow response to these kinds of species is absolutely deadly in the sense of enhancing their potential to spread. And once they spread, too far, then it's almost impossible to put the horse back in the barn, sort of thing. So those are two elements. Natural spread rates, I don't know enough about that to actually compare them, but to some degree it depends on how far these insects will fly, and we know that ALB has a tendency to stay close to its tree that it originated from. So typically they'll emerge, feed in that tree, and then oviposit right back in the same tree. And that might be a benefit in terms of being able to control that species a little bit more easily than EAB, which tends to fly further and infest new trees close by.
Next question here. How do you deliver the fungi that you use for controlling Asian longhorned beetle?
Okay, so I want to emphasize that that's not our work. I have done no research on fungi. Most of that work has been focused on using these impregnated fungal bands. So it's like a fabric that is used, deployed around the tree, and that fabric is impregnated with the fungus. The reason they use those bands, by the way, is because it enhances the longevity of the fungus and allows it to be effective for a longer period of time. And for example, if you spray the fungus straight onto the bark tissue, as an example. There's actually been, so those studies are done by Hajek and her group at Cornell University largely, but there's been other studies on other fungi as well, including Bovaria. And I should just point out that there's more than the work that I emphasized there today. So Dubois, Shanley et al., and others doing work on that fungi. And if you Google that on the net, you'll see lots of references to that sort of an approach.
And can you give some examples of how pesticides were used against Asian longhorned beetle in the U.S. or in other areas of Canada? So was it used in combination with tree removals, or how was it used?
Yes, so all of the operational use in the U.S. so far has been with imidacloprid only, and it's always been used in conjunction with an eradication attempt. So it's an integrated sort of approach, and it's really, you could think of it as being used to assist the uncertainty associated with where the population is, the infested trees and the susceptible host trees. So that it always is being used or has been used in conjunction with destructive removal. And you could think of it as basically putting a chemical buffer around that area where they believe the infestation has been, and trying to ensure that anything that's missed will be killed by the chemical buffer around those areas. A good example of that has been in the Chicago infestation that is now declared, or has been for many years now, declared eradicated, and imidacloprid was used as a chemical buffer in that eradication attempt.
Okay, I think we just have time for one more question here. So when you use TreeAzin to control the Asian Longhorned Beetle, will it have to be applied every second year, similar to emerald ash borer, or is it applied at different rates and different times?
No, I can say that we will have to do a double application of TreeAzin, and that's because it's non-persistent. So that compound degrades naturally within the tree through time. And so if you want to induce high levels of control over a population and make sure that you get the various life stages, and particularly in a case with ALB where they're protracted emergence and so on, you'd have to do at least a double application. And the difference might be that a strategy for ALB might differ in the sense that we could apply 2 years in a row rather than every other year as we do with EAB. And the reason you might want to do that with ALB is because of that protracted emergence of the adults. So if you did it 2 years in a row, you'd be effectively ensuring that both the larvae and the adults of those years are going to be directly exposed to sufficiently high levels of TreeAzin to induce control.
Okay, great. Well, on behalf of everyone, I would like to thank you, Dean, for joining us today and giving us some insight into the research behind the control of Asian Longhorned Beetle in Canada. I encourage everyone to register for our next webinar, which will be held on Thursday, April 23rd at 1 p.m. Next week, we will hear about the hemlock woolly adelgid. Affecting the ecologically significant hemlock tree, the hemlock woolly adelgid threatens biodiversity and wildlife habitat. 2 detections have occurred so far in Ontario, one in Etobicoke in 2012 and one in Niagara Falls in 2013. Hemlock woolly adelgid continues to spread across the northeastern United States, and additional introductions into Ontario are likely. We will hear from Dr. Mark Whitmore from Cornell University on the control efforts occurring in the United States. Thank you to everyone who logged on today to view our presentation. You can visit our website at www.forestinvasives.ca for more information and to register for the rest of the series. I hope to see you all back here next Thursday. Thank you and have a great afternoon.